here. It's not as much of a battle as I've heard it is in other states. So
that's good.
Yeah. How's Iowa then?
We have a mixed, some classes are in person. Depends a bit on the instructor. But yeah, the large groups have to be online. And then labs, some people converted labs into just virtual. Others of us have continued to do labs and just spread people out or have some system to, offer some protection. But we just heard that in the fall, we are expected to go back to in-person all activities.
Yeah, I think that's the goal here as well, is they want to, maybe with the exception of the gigantic lectures. My genetics class is going to be 220 students, and so there isn't even an auditorium that we could space people out far enough, so they may make another section or offer it hybrid. We'll see. It just seems silly to stuff that many kids in one room. Honestly. Yeah. It's not a great idea.
Air quality is going to be poor anyway. Well, it is 3:11, so I'm going to welcome everybody to seminar and introduce our speaker today, Dr. Allison Oakes. She is currently a postdoc fellow in the Department of Environmental Biology at the State University of New York College of Environmental Science and Forestry. It's a huge name, but
it's the biggest, actually. It's the largest.
Okay. It's maybe more often known as the SUNY College of Environmental Science and Forestry. So those of you in that field probably are familiar with that. And Dr. Oakes has gracefully agreed to talk to us about a project on American chestnut. It's part of a really interesting, fascinating project. Success story on American.
My computer is being a little weird, so I guess I'm just gonna listen to the lecture on here.
Oh, okay. I'm gonna ask the audience if they can all mute them. So everybody's muted now. Okay. And so Allison, Dr. Oakes works with Dr. Bill Powell and his team, right? And I'm sure we'll hear a lot more about the project now. So welcome, Allison.
Well, thank you. Thank you very much for having me here. It's always exciting to kind of share the cool things that our project is doing. So I've been with the American Chestnut Research and Restoration Project since 2006, and have seen that was the year they first planted a transgenic American chestnut outside in the ground via permitting. And so it's been just a fantastic ride. It's been a great project to work with, and we've gotten some really fascinating results in the past few years. So here we go. We'll kind of dive in. Oh, let me find I had Zoom. Sorry, I got to get back to there we go. Okay. So this is not to be confused with the horse chestnut, which is in the soapberry lychee family. So American chestnuts are in the Fagaceae family. Okay. Castanea dentata. So it's closely related to oaks and beeches. It's a pretty fast-growing hardwood. This particular timber type is what we're really interested in terms of getting trees back out into the forest. It grows extremely tall. It needs to break canopy in order to flower. And then it got absolutely massive. They were called the redwoods of the East. And you can see a 6-foot lumberjack there for scale in this photograph from the early 1800s out in an American chestnut grove all along the East Coast and up into in the Appalachians and down into Georgia and Mississippi, as we'll see. Just an absolute gigantic tree. Okay. And it pours just mass down every fall. There are accounts of there being chestnuts and burrs littering the ground up to 6 inches to 12 inches deep in places, just all across the forest floor, a massive amount of calories. And the wood was very rot resistant. It has very high lignification, and so there's many old structures still around, like cabins and barns, that are still standing after hundreds of years, just due to the how long lasting the wood is. And humans made a ton of use out of the chestnuts as well. In some Appalachian counties, the main food export or crop export for the entire county was the sale of chestnuts in the fall to the cities. For chestnuts roasting over an open fire, which were a holiday treat throughout a lot of the eastern US. It also makes a great gluten-free flour. I've baked with it. It's fantastic. Adds like a nice nutty flavor. You can order it on nuts.com. It's not going to be from American chestnut. It'll be from European or Asian chestnuts. Very, very nice flour to work with, and especially makes great beer. I don't know if anybody else likes beer, but it's beers and liqueurs. It has a wonderful nutty flavor that is somewhat reminiscent of hazelnut and pistachio. But the main contribution to these nuts was mainly eaten by all the critters that live in the woods, from bears to deer to turkeys to passenger pigeons. They hypothesize American chestnuts are quite small compared to other chestnuts you might see, and were hypothetically a big component of the passenger pigeon's diet, which when combined with loss of habitat and overhunting likely led to its demise, along with a lot of other animals that were uniquely dependent on chestnut. And there's a fish there because leaf litter is extremely important for aquatic ecosystems, and chestnut leaves just were one of the most massive components of that leaf litter up until relatively recently. So there was definitely an impact on stream invertebrates when the chestnut kind of left the scene. So speaking of leaving the scene here, here's the range of the American chestnut post-introduction or pre-introduction of the chestnut blight, down into the highlands of the Mississippi, Alabama, up into southern Ontario. There's a relatively large population, and everyone from Maine was very excited to tell me that that Maine map is wrong, and there's a lot more American chestnut in Maine than just shown on that map there. But pretty much the entire range of the Appalachians was home to these trees. So what happened? Well, the chestnut blight, a necrotrophic pathogen, Cryphonectria parasitica, came in on Chinese chestnuts that were being shipped to New York as decorative trees. And they were carrying along a fungus that they had become co-evolved with and were resistant to, but what their cousins, the American chestnuts, were completely unresistant to. So what happens with this tree is that the fungus enters through a hole or some sort of wound in the bark, and then it just spreads rapidly all the way around the tree, completely girdles whatever stem it's on, and kills the chestnut back down to the ground. Now, the saving grace of the American chestnut is that it does sprout from the root crown. We get lots of stump sprouts off of it. So even if the rest of the tree dies, it can sprout from the base. The problem being, those sprouts can never get big enough to breach the canopy. They'll eventually get about 3 or 4 inch in diameter. The blight will reinfect a wound and it'll get killed back down to the ground. So it's this like Sisyphean battle of rolling uphill all the time. And so this spread very quickly throughout the range. They tried fungicides, they tried cutting down every tree in like a 10-mile radius. There were all sorts of things that foresters tried to do to halt the blight, and none of them were successful. The blight is blown around on wind via spores. It will grow on oak trees, not harming them in any way, just saprophytically on the bark, and it will linger in the environment for quite a long time. So some people this reached the Ozarks in about the 1950s. There are people alive that remember the Ozark chinkapin, which is also kind of a forest tree, which also got attacked by the blight. But they actually remember going out and like gathering Ozark chinkapin nuts with their grandparents and such, although we've kind of lost that in our area in New York State as it arrived in the very early 1900s. Okay. So this is a chestnut forest in spring, right around early July there. You see the flowers just look like snow on top of the trees. And they were estimated in some areas as much as 25% of the tree cover was American chestnuts at this time before the blight came through. And then when it did, you can imagine if 25% of your local tree cover up and died, similar, maybe some people remember when the American elms died, when Dutch elm disease came through. It's just losing that sort of biomass has a massive effect on the ecosystem. So people have been trying to fight the blight or save chestnuts for over a century now. So there's multiple pathways to go and some different choices for restoring the tree. There are chestnuts on the market. You can buy Dunstan chestnuts, you can buy Chinese chestnuts, you can buy Japanese chestnuts, all sorts. And so the Dunstan chestnut is very popular. One of the things they have it for is for deer. If you want wildlife to come to your property so you can hunt, that's great. It works perfectly great for that. Problem being some species, so here are some mallard ducks eating American chestnuts. You can tell the burr size is much, much larger on these Asian variety chestnuts. And so some species are not actually able to eat those. And they're not, Dunstan, a lot of these hybrid chestnuts are not true to type. They're not like cloned. Chestnuts are very difficult to clone and propagate. So they're usually a seedling allotment. So if you want the same type of tree every time, you're getting, it's kind of a grab bag, but they're great for orchards. They're great for wildlife. If you just want to have chestnuts to eat, that's awesome. But these Asian forms are pretty short trees, maybe like 40 to 60 feet. And what we really want for restoration are those massive American chestnut giants with the small high protein seeds that would work in our, and be able to succeed in like a shade tree forest, which these don't. The hybrids do not survive well at all in shade tree plantings from what we've seen. So we don't think the hybrids can really replace the American chestnut. And so has been a lot of work on back crossing so that the hybrids are more and more and more like American chestnuts. Okay. Oh, there was a radiation mutational breeding that tried as well. And some of those trees are still around. No, they did not magically mutate and become resistant to blight. But that was a big thing in the 50s and 60s was mutation. So the American Chestnut Foundation that we work really closely with has been working on the backcross breeding program for decades now, with the idea that, well, what if you could get those genes that you want, and then you could breed back to American, breed back to American, breed back to American, and get a very, very American-like tree that would just have the genes of resistance that you want. This plan would have worked if the initial hypothesis that there were only 3 resistance genes was true. With more genetic testing, we found at least 9 different loci and probably 18 or more genes that are all involved in the Chinese chestnut resistance to blight. So breeding in 18 genes is not going to work so great, because as soon as you breed it to a completely unresistant, like locally adapted, American, you're going to lose a bunch of the genes that were all building that quantitative resistance, right? So there's still very excellent trees. There is hope because there's another pathogen coming along, Phytophthora, that appears to have a much smaller quantitative resistance, maybe only 3 to 4 genes. And so this backcross breeding could actually be really useful for incorporating the Phytophthora resistance with a chestnut blight resistance. Okay. It's also tricky when you think there's 40,000 genes at play in both species, and getting the right complement would be a very luck of the draw type thing. Yeah. So I can skip a little of this because you guys are all biologists, with the knowing that gene is like a recipe in a cookbook, and a chromosome is a shelf of cookbooks, right? And so a whole genome is like a library of cookbooks that you need to go into and find the right things. And that with breeding versus transgenics, what we're looking at this giant pool of 40,000 gene pairs in the Chinese and the American. If you look at a back cross, even the furthest back cross they've gone, you're still replacing 1/16 of your American chestnut genes with Chinese. So that's almost 2,500 genes that have been swapped between. So if you take a regular paperback book with 200 pages, that's like removing 10 pages of text and replacing it with maybe the Chinese characters. The story could still make sense. It depends where the different words got switched out, how dramatic the change was. Was it a whole paragraph, or was it like the key suspenseful moment of the novel, or did it not really matter? It's kind of different for every cross you do. The idea with genetic engineering is that we can be extremely precise, and that we're not removing anything when we add something. So the idea of taking an entire book and inserting 2 words without breaking, replacing, or changing any of the words, that can happen, but we certainly wouldn't move forward with anything that broke up the meaning of a very important sentence or something. Just to put the amount of change between a hybridization program and like a genetic engineering program in perspective. We want to work at these minuscule targeted levels of change. So what gene do we test? from Asian species, other plants. We did do some testing with some of those 18, what we call cis genes from the Chinese chestnut that could potentially be bred into American chestnut. And so far the results with that is that we get a very slight increase in resistance for each gene added. And then you're up to the same problem again of now you got to get 18 genes in to get up to a comparative resistance with Chinese chestnut. And that's currently not feasible with our technology at the time. So we looked at other plants and we found this gene, or Bill found this gene in wheat called oxalate oxidase. And what it does is it simply takes oxalic acid and breaks it into hydrogen peroxide and carbon dioxide. So the nice thing about this isn't a pesticide. It doesn't kill the fungus. All it does is break down the fungus's main weapon. The Cryphonectria parasitica releases oxalic acid to lower the pH in the cells around the wound site. And then as the cells die, it can nibble away at the dead cells. So if you break down the acid, the fungus sits there and puts out more acid, and that gets broken down and the fungus sits there. And so we're not killing the fungus, we're just removing its weapon. And this also reduces the chance of the fungus developing resistance to our gene here, our oxidase, because it can go through its full reproductive cycle just hanging out on that small little wound on the tree. So, and then instead of killing the fungus directly, we're just changing its lifestyle from a pathogen to saprophyte. So the two species are still living together. The fungus is still colonizing around that initial wound, but it's not growing further and destroying the entire cambium of the tree and killing it down. So oxalate oxidase is very widespread in the plant land. Wheat is the one we use because it's the most studied, but it's also in corn, barley, rice, okay, grains. It's in peanuts and strawberries, walnut, tomato, cacao, sorghum, potato, apricot, pea, dates, oil palm, beets, and that's just the ones we were able to find references to. There's probably a lot more. We have found one in our native black walnut that's a pretty high match that has an oxalate oxidase enzyme. But again, we worked with the wheat one because that's the one that's been most characterized and we know so much about. So it's also nice because we have a very quick assay. You don't have to do PCR to find out whether or not the gene is in. We can do this just, at a glance, qualitative assay that here's banana with oxalic acid. And when you put the oxalate oxidase solution on here, you see the staining if it's present. So this is one that doesn't, this is the negative control that doesn't have the oxalic acid. And this is the one that has it added. So it's a nice way that we can, if somebody wants to know, is a leaf from this chestnut tree on my farm, is this a GMO chestnut or not? Very simple. We hope to eventually be able to make these kits a little easier and faster, but you can tell whether or not it is transgenic or not based on a simple assay. So next step is, how do you get a gene into a plant? I'm sure some of you know this quite well. It's very easy, just a couple of steps. Clearly nothing too long and intensive. Anyway, so first you choose your gene of interest and you prepare it in your agrobacterium. Agrobacterium being a natural genetic engineer, it's going to, under proper conditions, insert its tDNA into your plant cells of choice. In our case, we end up using immature embryos. So we have to first isolate those. We have about a 2-week window and it's got to be perfect. So you can do this for days and days and days, and it may not work at all, but you eventually get a sweet spot. And when you open up those tiny little burrs and you pull out the tiny little seeds, you can cut those open, and we do a lot of work in laminar flow hoods here, and extract those immature embryos. This one's a little late because we see one embryo is already starting to take over from the rest, but usually be when these are all about the same size. Pull those out and start culturing, and we have about a 1 in 1,000 success rate for this particular procedure. It is very time and labor intensive. But once we have a few somatic embryo cultures going, we can grow them up on a plate, and we can take our prepared agrobacterium with our gene of choice, literally smoosh them together for a while, and then we go through selection using a ebb and flow bioreactor to kill off the agrobacterium and any transformed tissue, and you'll eventually get American chestnut embryos that have your gene of interest and are expressing them. And that's the first part. Then we go and check and make sure via PCR that those embryos actually have the gene of interest. And then we end up taking those little embryos and putting them on a successive levels of plant hormones to convince them, stop being an embryo, now be a shoot. Hooray, plant tissue culture and its marvels. We get these little teeny shoots popping up. And then we can take those and move to shoot culture, make more, and check them for gene expression and copy number. We want to know that the gene hasn't been inserted too many times. We're looking for single, possibly double copies, but just one insertion would be the best. And then to see if those new genes are actually expressing. So if the oxalate oxidase is actually being produced. Yeah. And then once we have shoots, we multiply shoot cultures up, we root them, which was my PhD work, was getting these little guys to root because they really don't like to and have to be convinced in various different ways. And then we have high humidity growth chambers that we use to acclimate the plantlets to lower and lower humidity over time. And then we used to use plastic bags, and it is a relief to get away from those. And then we get these lovely growth chambers full, and then finally greenhouses full of transgenic plants that we can take out to the field and start in field trials. So, and then we check them again. We actually do a leaf assay that was developed by one of my coworkers, Andy Newhouse, to check to see if there's any resistance to blight. So we're trying to get away from large stem assays to small stem assays or leaf assays, so we know earlier and earlier in the process, do these plants have resistance to chestnut blight? So here's a preliminary, one of the very first lines we made. You can see over on the left is the American chestnut control with the sunken canker. It's gotten very large very quickly. The Chinese resistant control, where the canker has stayed small, contained, hasn't grown a lot. And then this Darling 4, one of our first events containing oxalate oxidase, where we saw the canker is smaller. It is smaller compared to the American and the Chinese. Now this had a vascular promoter attached to the oxalate oxidase, so it was only expressing in the vascular tissue as opposed to a constitutive expression, which is what they tried next. Is what if we use a constitutive promoter? And we found that as we produced more oxalate oxidase, as we increased the transcription of that gene, we saw the necrotic area in the leaf assays going down. So the smaller and smaller the cankers were getting as more and more oxalate oxidase was produced. We're actually getting to the point that we're slightly better, more resistant than Chinese chestnut to the blight, which is helpful. So here's another kind of one of our small stem assays where all of these are infected at the same time, and they're different heights because we went for Chinese chestnuts get thicker stems faster, so they're a little shorter. But all the American chestnuts died back after this. You can see they're sprouting again from the roots, as they are wont to do. All the transgenic trees in the middle survived, and then we actually managed to kill off half of the Chinese chestnut with this particular resistance assay, but promising so far, even in the greenhouse, showing that they've got this tolerance to the pathogen. Now, the big key issue, is this heritable, or is it going to get silenced or shut down? Yes, it is heritable. That's one of this paper was about tracking through to the T1 progeny, whether or not the blight resistance was getting passed down. The idea being it's sort of a simple Mendelian dominance recessive issue here. If any tree with one copy of that gene for oxalate oxidase will show resistance to the Pryphonectria parasitica. We're down to, I think we're planting out, we've done T1, T2, and we're planting out T3, and we're not seeing a degeneration of the resistance effect. Half the offspring don't have it. If they don't have it, then they are fully resistant, but if they do have a copy of the gene, then they do have this resistance. It worked out very nicely. And so another key technology we found was that if we could convince chestnuts it was midsummer in July and that they should bloom to save their life, then they should. So we have these high light growth chambers, just really pumping photons on these guys, and you can convince them, and these are less than a year old, to flower. So one of the big problems with the breeding project was that you had to wait like 7 years for these things to flower. Maybe if they're in full sun, you could get some male catkins, and maybe 5 or 6 years. But to get female flowers, you had to wait more like 7 or 8. So with this fast flowering, we can at least get pollen and take this pollen and cross it and get a nut in the fall, and then grow that nut up in the greenhouse over the winter and get pollen the next year. So shortened our breeding cycles down to 1 year. So we can do a generation a year as opposed to waiting 5 or 7. They look like big giant fireworks here. They're just beautiful flowers. And then we can, one of our graduate students has been working on how do we store it, how do we maintain viability, how do we test viability. And so we have gotten to the point we are confident in shipping pollen to people that want transgenic chestnut pollen. If they say had their own trees in the future when this is available, we could send them pollen and they could actually pollinate their own locally adapted survivor chestnuts to have offspring that incorporate the genetics of both the local adaptation parent and the transgenic parent. So it's easy enough as just brushing them on glass slides. The static electricity is enough to pull the pollen off, and we put it in a little slide box and we're good to go for the next year. So these are the female flowers, by the way. And there's the male over here and the female over here. And this gets fun. We get some very strange looking trees over the summer, as anything with transgenic pollen needs to be bagged so that it can't escape. We're under permits via the USDA. We have very strict regulations. All the fields have to be fenced and locked, and all the transgenic material must be contained. Just like the transgenic pollen here has to be in a bag. And then over this bag will go the squirrel-proof bag, because squirrels make very quick work of a cute little paper bag. So we get a whole like screen netting bag over top of these. So we rent lifts a lot during the summer to get this done. And then at the end of the summer, when everything's gone, we just nip the branch off. Here's Bill cutting off a full batch here. And when we take the bags off, we collect these burrs, these huge spiky burrs that break open. And that's the other reason to have the bag, so no possible transgenic nuts are lost. We haven't lost any so far. It's been good the past like 5 years. So, but we'll open up the nuts and then they get individually, we've labeled all the pollen bags with which crosses which. So we've got all these fancy pedigrees for what the backgrounds of each of the lines. And then a lot of the nuts are not viable. They're sort of shrunken. That happens with any pollination, transgenic or not, but we sort of go through, get rid of the not viable nuts and then grab all the viable ones and pack them into a little baggie with some moist peat moss and germinate them for the winter. So here's a lovely burr with 3 nice round fertilized nuts. Yeah. And then it's really fun when we get a whole bunch of them. They're soft and velvety and they're delicious. If we have extra that aren't transgenic, we occasionally will test out to make sure they're still, tasty. And then all those nuts get planted and assigned an individual number that we can track back to who their parents were and put out in the greenhouse and get grown up ready for planting out in the fields. So planting days are huge. We, bring friends, we bring kids, and it's just a really great community event to get out there and plant trees all over. And so it's quite fun. And then we sit back, wait, watch. Our talented field team takes care of all their little tree wants, and we get these lovely tall trees. And so getting back to the transgenic part of it, what we'll do is we'll put out offspring, we'll put out siblings that we know have the gene and that we don't. And then when they get inoculated, it's pretty dramatic distance. So here's difference. Here's a sibling that had a non-transgenic mom, a transgenic dad, and this one did not inherit the genes for resistance. This is the sibling that did. Just by having one copy of the oxalate oxidase gene, the infection is contained. The fungus is, you can see the little orange there, it's producing both, we've checked its sexual and asexual spores. It is going through its full life cycle, but it can't get a hold. It can't rip the tree apart with that oxalic acid. So this is the goal here. You live in peace on the tree and don't kill it, and we'll be fine neighbors. So the idea now is to incorporate as much genetic diversity as possible. And this is the Mother Tree Project, where we're working with the New York State chapter of the American Chestnut Foundation, is to outcross the transgenic trees to as many regionally adapted surviving American chestnuts as possible. Because you're going to get a different complement of the maternal genes and the paternal genes with every offspring you make. They're not all going to be the same, just like siblings get on average 50%, you know, from mom and from dad, or from paternal, maternal grandparents, right? So the idea here is to rescue genotypes of these locally adapted trees so that they don't, when the trees eventually do succumb to blight, that we don't lose that genetic information. Outcrossing is the name of the game. So eventually we would want to make trees that always give blight resistant offspring. By, if you mate two heterozygotes together, you get a 25% chance of getting that dominant homozygote that would give then, every time it mates with anything, all of its offspring are going to have the transgene. But we don't want to do that until we've really maximized outcrossing and really good genetic diversity. So, well, what about, okay, so you're putting these trees out there that break down oxalic acid in their tissues. How does that affect everything else that it's going to encounter in the ecosystem? And we have done a whole bunch of studies on off-target effects. EPA is very interested in that. So what about mycorrhizal symbiosis? Do we somehow mess that up? Is that something other mycorrhizae are using as oxalic acid? And so a grad student did look this up, planted a bunch of seedlings, transgenic, non-transgenic, and other things. Looked at, they morphotyped all the ectomycorrhizae, and they did a bunch of DNA analysis, RFLPs, and found that what really mattered was the soil type. She did this in 2 separate plots, and one of our plots had much better colonization than the other. And so over here is our wild American and our transgenic American, showing percent colonization of root tips, and there wasn't a significant difference. And so the other ones here are American beech, red oak, Chinese chestnut, and hybrid, F1 hybrid chestnut were the other, but we did not see anything like the transgenic American having less, had more of it. And then how about amphibians? You know, leaf litter is an important part of streams, and is there going to be an issue if transgenic American leaves fall in streams and then say amphibians are nibbling on those leaf shreds? And some poor undergrad had to change water on these, you know, once every 3 days. That's what undergrads are for, exciting things you do in scientific research. And one of the things we looked at was the Gosmer scale, the how amphibians develop over time, and basically found there was not a difference between any of these different leaf types. We had the non-transgenic, transgenic, hybrid chestnut, Chinese chestnut, sugar maple, American beech. They really didn't like it if we didn't give them any leaves. Okay, so we're not seeing a difference of tadpole development there. Surprisingly, when we looked a little closer at the ones that we didn't give a food supplement, if we just gave them leaves, how did they do at 8 weeks? And surprisingly, both the transgenic and the non-transgenic chestnut, the amphibians did the best. They had the most development at that time. They did worse on, significantly worse on hybrid chestnut, Chinese, sugar maple, and beech, which kind of makes sense if you think wood frogs, you know, have been living in the Appalachians for, you know, thousands, if not millions of years. I don't know the phenology of wood frogs, but they've co-evolved to eat chestnut leaves as the predominant source of leaf litter. So it's interesting seeing this play out, that putting out the transgenic chestnut would possibly help you know, other species that were having issues over the, well, their food source has been gone for over 100 years now. How about bees? Pollinators, hot topic. What if we put out something that hurt bees? Bad idea. Nobody here wants to do that. So we got our own beehives, which we have been making chestnut honey for the past couple of summers from the beehives, so that we could recruit bees from them. And we collected the little pollen nuggets off of the bees as they fly in. You can kind of knock a bit of pollen off their knees when they're flying in. Not all of it, we're not going to starve the bees. And we get these little pollen grains. And so what we did is set up these micro colonies with help from entomologists at Cornell University, and looked at, all right, well, what if we give them some oxoenzyme in their nectar, in their pollen? How will that affect their survival rate and offspring and everything? And so you see the little bees, if you get 5 bees together, they start to act like a hive, and they'll start building egg cells and depositing larvae in there. And here's the survival. So they don't do super great in captivity, but we want to see, do the ones that we give a lot of oxoenzyme, the high rating here, does that change VM, we saw doesn't look like there's a whole lot of difference if we don't give them any, if we give them some, or if we give them a lot of enzyme in their food. And surprisingly, hive source mattered a lot more. We had a lot that our block there sort of made a huge deal in survival. So generally one, whether or not a hive is healthier or not made a lot more difference in survival over time. And we didn't see any difference in reproduction or other factors that we looked at, pollen use or how much nectar they drank. Everything came out even. So we have not found any off-target effects so far. There are many other studies we've done beyond this, like surrounding vegetation. Somebody's doing deer herbivory this summer, insect herbivory, and allelopathic effects of chestnut leaves, and we're just not seeing it behave any differently than a regular old American chestnut would, other than it doesn't die from blight. So that's exciting so far. And meanwhile, we're getting these really lovely orchards of the trees, and we're just watching them over time. We're looking into, like, how about photosynthesis? Is there a growth or metabolism effect of producing all this enzyme? So there's a lot of neat ongoing studies, and we're seeing a lot more media interest in chestnut as a genetic engineered organism that's not for profit, that's for conservation, that's for ecological restoration. And the people who are, I've had friends that are completely anti-GMO be like, except for you, I like the thing you're doing with the chestnut, but Monsanto is terrible. I'm like, we can talk about Monsanto is a totally different topic, but thanks for chatting. So one of the things we're doing, as anyone who has a genetically engineered organism needs to do, is talk to the federal regulatory review people. USD APHIS being one, the FDA, because chestnuts are edible and people are going to be eating them, and the EPA, which is worried about, like, is this a pesticide, is this insecticide, is this mitigating a pathogen? And so we're on this route. We have submitted to the USDA. We've gone through our open comment review. We currently have drafts for both the FDA and the EPA. But they want to hear back, see what the USDA says before they want us to submit. So it's kind of a massive loop. Current plantings that we have are still, again, all permit only. Everything is under USDA permits, no accidental release, but everything has to be mapped. They come out and make observations. We have to report any change we make to the plots. Our sites get inspected yearly. And we do have an amendment that we are starting to possibly have some limited open pollination of the chestnuts based on our research to this point. So, but the monarch butterfly here is enjoying some chestnut pollen, non-transgenic. None of our transgenic trees are quite that big yet. So I don't think I'm running a little short on time, so I'm going to skim through. The USD really wants to know, are we a plant pest risk? Are we somehow making an American chestnut that is weedier or more invasive than a regular American chestnut would be if it wasn't dying from Cryphonectria parasitica? So that is underway. And then we have the FDA. They really just want to know, are your chestnuts significantly different from what's on the market? If you could buy chestnuts, I love the chestnut packaging here using a horse chestnut leaf, which is definitely not edible on their chestnut packaging. Good job, marketing. But basically we've done some nutrient analysis comparisons, and we're right on the money. Like, it's basically the same amount of carbohydrates, proteins, vitamin C that a regular chestnut has. So that's what they want to know. And then the EPA wants to know, is this a pesticide? Because technically this enzyme is breaking down something and adversely affecting the growth of a pathogen. So we need to discuss whether or not we're going to have a registration, or are we going to have an exemption? But there's a lot of that in the mix. We're actually working with a couple NGOs that know how to speak the language of the regulators as opposed to how scientists view things. There's certain things they want in their giant volumes of documentation. So that's ongoing as well. And the whole thing is this idea of the coordinated framework is that there's just you know, it should be risk-based and product-based. Like, is this particular thing that you're making an issue, as opposed to looking at how did you make it, the technology, you know, was it mutation bred, was it hybrid, was it genetically engineered, was it CRISPR engineered, but just what is it now and how does it actually affect people in the environment? So it's fascinating, and definitely something that we've been focusing on the past year or so, as well as other plants. So there's a lot of preparation. It's a very long process. So the response so far has been, the regulators have been encouraging. The public opinion has been overwhelmingly positive. Like, there have been some, you know, threats and negative publicity and such, but for the most part, people like to see a tree being developed to help the ecosystem. And we're not patenting. The college is not going to be getting a patent. I think we're going to trademark just to protect the variety lines for growers who want to grow these types of trees, but no patent. And the idea is the tree will be open and available to the public, you know, hopefully, and there's plenty of orchards that are interested. So the idea being, well, the chestnuts are the first, they're paving the way. What other trees could possibly benefit from this? And then there's, so, you know, elm, which I'm working on, American elm. There's the ashes, the walnuts, western pines. There's plenty of trees that need help. And so we're only working on chestnuts, chinkapins, and elms. So far, I know Purdue has the ash program. I don't know if anybody's working on walnut right now, but there's a lot of definitely ideas there. There's chestnut is a success story to sort of bolster other things like corals that can survive warmer ocean temperatures, or a lot of endangered like island species, like in Hawaii in the tree there, the Ojai tree, and that we're really kind of paving the way or opening the doors for genetic engineering to be considered a potential tool for forest and ecosystem health if applied in a responsible manner. And it's not like this was done overnight. I mean, we started in 1990 is when the TACF reached out to Bill and Chuck and said, hey, could we do this? You know, it took another 7 years to be able to culture embryos and regenerate shoots, and then took another decade to get a transgenic chestnut planted, and then finally we could show that they are resistant in 2012, and then we could show in 2014 resistance is terrible. These projects take a long time. Trees depends on the tree, of course. Chestnut's pretty recalcitrant, but this isn't something you just like, oh, I'm gonna do this overnight. So what's next? We're looking at how these trees grow over the natural range. We're collaborating with Penn State and Virginia Tech. We have plots of trees at different latitudes now and look at, they're being planted so they can grow very large. We can start looking at the long-term environmental impacts. We want to see how they, you know, vernal pools and other things work with chestnuts. And so we did a bunch of plantings in 2019. 2020 was a little weird. We're getting back to planting this year, and so it's going to be very exciting. And just and the next step is I'm looking at, I've got a postdoc fellowship. I'm working on transforming American elms for resistance to elm yellows, a nasty little bacteria there that is hot on the heels of Dutch elm disease as a big problem. And so we're looking at that from a interesting, using trees that have been bred to have Dutch elm resistance and then genetically engineered to have elm yellows resistance, and what sort of neat things can you do when you combine forest health tools together. So I'd love to take any questions you have. I hope I didn't run too far over, and thank you.
Allison.
This is my first presentation like in months, so apologies if I'm rusty. So
it was great. So we have plenty of time for questions. So who has questions for Dr. Oakes?
I have a question, Leonor. Thanks, Allison. That was a great presentation. I really enjoyed it. So I had a question about, you were showing the transgenics seedlings in the nursery, and you did a comparison between the two siblings.
Yes.
Where, so we had one with the canker on the side, and then the other one, the one with the transgene in it, much smaller canker. But you mentioned there that there was a little bit of orange there, and so the fungus was going through its full life cycle. And so I wondered if there was any concern there about developing resistance, because you are going through the whole life cycle. And so, you know, and so if you're getting sexual reproduction there, could that lead to resistance to that oxalic acid?
I mean, the fungus would have to start producing a completely different acid in order to overcome that. We've seen variable amounts of acid produced by fungus, and what we do, we use the most virulent lines when we test, so we know that we're, you know, really hammering the trees with something difficult. And so those were, since even a not very virulent strain can reproduce, it's not going to select for the virulent ones as much. Maybe slowly over time, but then you're going to see the trees taking over. At least they have something to improve upon, and natural selection is going to work on the trees in the opposite direction. You know, the ones that produce the most enzyme are going to be, you know, most likely to survive. So, but overall, we haven't seen the virulence of the pathogens changing over time. So,
thank you. That it's really cool work.
Say, Allison, this is Tom Harrington. So have you seen any natural infections of your control trees in any field plantings?
Yeah, we lost a bunch of our really champion mother trees over the past 2 years because they are just the non-transgenic ones that we use for their the female flowers, because we have older trees to get the female flowers, but they're all just dying of blight. We had to take a bunch of them down. We're currently short on mother trees because of how ravaged the plots are. Not even from like the infections that we do for inoculations, but you're right, they're just getting natural infection all the time. We're surrounded by oak and beech, and the stuff we're putting on the trees is staying there. We're not culling the trees out. So yeah, we've got a pretty high intensity blight infection in the orchards, but some of the trees are just thriving, and we're trying to keep all the other ones that aren't resistant alive in order to get more mother trees down the line.
But what about the younger planted ones? Are you seeing cankers on those and not in the GMO chestnuts?
They're kind of have juvenile resistance. It's not until you get about yay big and the bark starts to crack a little bit, and you get the fork, cracks in the forks of the branches. Yeah, they don't really get infected until they get old enough to have that like ripply bark.
So you really haven't seen that stage yet when they become.
We're starting to, now that we have the limited pollen release, because before, like we would have had to climb up the tree and cut off every single catkin. And we're like, we can't do that. We just have to cut the tree down to the ground, let it resprout. And so we could never see what happened when they got big, because they were always had to be cut back. So now we are starting, that's one of the things we're looking at like this year and at the brag plots, is how does natural blight infection look on these, comparing the two on older trees. Yeah.
Okay. Thanks.
Allison, a question. In your elm yellows work, are you looking at a super long time horizon there too, or is that something that you can see some lessons learned from the chestnut blight breeding work that would speed that up at all?
Oh yeah, it's I already have some transformed elms, working on rooting them now to get them planted this summer. So it's by far accelerated. I mean, all the protocols are in place. I can work from my colleagues who have developed the permits. So I have a framework to go off of there. We've got the infrastructure to handle massive tissue culture production of transformed plants. We've got the growth chambers and both high light and high humidity. We've got greenhouses that are set up for the care and planting of trees, right? And we've got a field crew that knows what they're doing with trees. Like, it's all there and ready to go. And that's sort of the big thing that USF is looking toward, is developing a native tree restoration center so that we can work with more, since we have all this infrastructure here, why not more species? And so that's the elm is sort of following the footsteps of the chestnut at this point. We're actually using some of the resistance genes that we found in Chinese chestnut that are analogous to Asian elms, and just sort of picking out a few that look really promising. Now that the transformation procedure is up and working, now the hunt for, oh, what other genes do we want to investigate for elm yellow's resistance? That's the big question now. We really had to get the transformation working first, but.
Thank you.
Mm-hmm.
Allison, I have a question too. So do you think there's the potential for this oxalate oxidase transformation to work in other crops with other pathogens that also produce oxalic acid, including maybe things that are more, annual crops, or that would be, the breeding process or the transformation process would be quicker? Do you think it could work with some ornamental or some other?
I think it's already being done against sclerotinia infections. Any pathogen that produces oxalic acid as a weapon, this would be a great tool to use for that particular infection. So somebody's like, put it in elm. I'm like, that wouldn't work. Elm doesn't have a problem with oxalic acid. So you'd have to, figure out which pathogens are doing that particular thing. And again, if it's a fungus that's secreting a different type of acid, well, then find an enzyme that breaks that down, and maybe you'd be good to go. So.
Interesting. Thank you. Any other questions for Dr. Oakes?
I'll ask a question. Just interested in, like, how many transgenic events, independent events, do you have to look at to find what you want? And then how do you decide, like, I assume if you're doing this FDA and EPA and tests and stuff, you need to probably pick one and go with it. So.
Right. Yeah, it's a lot of, that's hence the emphasis on outcrossing, because we're currently working with one event. We had, it's sort of, the field was much bigger. Eventually, in the beginning, we were co-transforming with GFP, to use GFP as our selection agent. And then we realized we're gonna have to breed out GFP. That's not a great idea. Like, what if, can we somehow just select for the one construct? So we went down to just its NTP2. So we have kanamycin resistance in order to kill off any tissue that isn't doesn't have the gene of interest. We had maybe like 15 contender events, but we ruled out anything that had more than one insertion site. So we wanted a single insertion site. And we got that down to 2, and then we found our darling 54, the insertion site was within another gene. So we excluded that one, and darling 58 was sort of it had one insertion site, and it was also in between genes in a non-regulatory region of just sort of junk DNA. We're like, okay, because we had to do the genome walking out from the insertion site to confirm that we hadn't sort of plopped into any particular specific area or gene. So it was a pretty high selection criteria to pick the one event that was going to go forward into regulation. And that's why we're so big on outcrossing, because we do just have the one event at the moment. There are more coming down the pipeline, different promoters, like a vascular and wound inducible promoter, a couple other ideas that we want to see. But for now, it's the oxalate oxidase with a constitutive promoter that is giving us like this really fantastic resistance. So
cool, thanks.
Yep.
All right, well, it's a little after 4, so I'm going to end the seminar and ask you all to thank Allison. We do some virtual clapping. Thanks so much for coming. And if anybody has any other questions, maybe you can maybe stick around for a couple more minutes after we finish the seminar, or maybe email her. But thanks so much for a very interesting seminar and for your time.
Thanks, Allison. Really inspirational.
Thanks for having us.
Thanks, Allison.